my laboratory was more focused on this Spartina. So when I actually started in 2004 at LSU, we did not have much of molecular information on this grass. So I was intrigued by its salt tolerance ability. So we were looking at to look at some genes that would be very highly responsive to salt stress in this particular grass. Although my job was also to look at the genetic diversity of this particular grass in the coastal states, in the coastal state of Louisiana. So there are several different accessions. So we used to collect and then do genetic fingerprint. So, but then because I was interested, so at that time, we did not have the high throughput sequencing technologies. So we used to still go. I used a technique called cDNA FLB, which is very small scale. Because whenever you deal with something with gel, so you cannot really think of in hundreds and millions and thousands. So we did, followed by that, we actually did a cDNA library of both leaf and root from this particular grass. So those cDNA were actually the one after actually exposing these plants into the salt stress. So they are salt-induced transcriptome, but mostly the cDNA library was prepared. And we actually identified a few of these ESTs which are very, very responsive to salt stress, whether it is in the leaf or in the root. Some of them we have, most of these are actually published work, as I said. So, I think Subho actually made me very clear that I should not be putting anything that is not published, since it is very open. And so this was actually kind of a very pilot scale or preliminary study of the biology, understanding the biology of salt tolerance of this grass. And then we actually leveraged this information to secure some funding, and then went for the genome-wide scan of that particular grass for the transcriptome profile. So originally we had a 454 that was actually the first generation sequencing, that is pyrosequencing by 454. And then later on we had added in 2018 we added actually the RNA-seq data also. So we have actually the 6x coverage of that grass transcriptome. And these are actually the, again, like these are the data. I'm not going into the details. We have now, this is actually the leaf, the transcriptome coming from the leaf, the unigenes, leaf under control and leaf under salt stress. And then this is root under control and root under salt stress, So we have a lot of unigenes that are actually common among the, between the two tissues and with and without stress, but we are interested actually the ones that are specific to the tissues and under a particular stress, under the salt stress. So whenever you have actually a RNA-seq or next-gen sequencing, you will get all this huge number of unigenes. And then what you do next? Okay. Before going to that, so again, this is a proof that using some of the genes, we know that this is a grass, so it should be actually close to the grass in the tree. And this is here Spartina, which is close to corn, GMS, and rice, and there is 105 viscosa, and potassium is over here. So as you can see that the Spartina and potassium, these two are actually kind of clubbing together over here. Okay, and then in the same clade is barley and then oats. And then these are, this is the wheat is over here, even though it's a grass, but then in terms of this genes for the salt tolerance, and that barley has also a lot of salt tolerance. So this is actually the other clade that contains these things. And Sueda, even though it is actually a halophyte or mesembryothymum abyssinium, because they are diploid, so obviously they have been in a different in the clade. And these are actually the genes of Spartina. They are actually distributed across the rice chromosome. You can see here. So these are the genes that are distributed across the the rice 12 rice chromosomes. Okay, so obviously there are some gaps, and we do have, this was actually made with only the 454, but these gaps we have closed down with the RNA-seq data, and then I have not included here that we have not published. Okay, and then going into the genes, then the objective is, when you have this next gen sequencing, the objective is actually to narrow down the genes that you really want to target. Right, that's actually your number one objective, that because you cannot possibly handle that many thousands of genes. So, and then you go for a gene ontology enrichment of these genes, and then you identify the genes or the processes which are actually enriched under salt stress. So, for example, here we have kinases and the genes involved in the transport mechanism, and the transcription factor like DNA binding or protein binding, and the genes in transferase activity or metabolic processes. So what the bigger the circle, that means more number of genes, and then the darker the color means they are highly enriched. Okay, so under salt stress. So we are actually targeting, that means this is a kind of a narrowing down of the genes, that you only select the genes that are in these pathways because they are more enriched under salt. And we actually divided that into leaf. I'm not again going into the details. So here, for example, under the biological process, you have more into the genes in response to stress, which is very expected. And again, in transport mechanism, those which are in the plasma membrane, we know that transmembrane genes with transmembrane activity or present in the plasma membrane are very important, because there are a lot of literature that actually suggests to that. So the same thing also for root. Again, I'm not going into the details of this. These are all published. But again, here also, there are also these genes that is very highlighted. They are actually more enriched in root cells also. Okay, so now we have these genes. So we needed to actually see their differential expression under salt. So the ones that you see here, the green dots over here, this is actually MA plot where you see the genes that are actually with the green dots, green circles. They are actually having log 2-fold over 2, which is, that means more than 4-fold increase under salt stress. Okay, and then the red ones, the ones that are actually down regulated, that means less than or 4-fold decrease in their expression. And this is actually just a snapshot of one of the couple of categories of this. This is actually the kinases, and these are the transporter genes. So as you can see that the blue ones, of course, are the upregulated, and the red ones are downregulated. And then with the hierarchical clustering, this clustering also helps us in, again, selecting out some genes or narrowing down the number of genes that we want to target later. Okay. So this is a validation of the genes, that what we actually see in next-gen sequencing, they are actually validated when we really do the temporal expression of those genes in both leaf and root tissues over different time points, very early to until 3 days. So we can see here actually there are differential expression, and some of the genes are maintaining their expression even at very late stages of salt stress. So those are some of the genes that we are actually also interested, because you obviously want to have a gene which is expressing, not only just expressing in the early stage. And some genes can express just because of a shock. But then the maintenance of the expression of the genes is very important, because then that these genes are actually truly responsive to salt stress, or they might be actually contributing towards the salt tolerance of this particular grass for their prolonged expression of those genes. Okay, and this is again the real-time PCR of some of the selected genes. In addition to the transcriptome, we also look at the small RNA profile, so that to look at the post-transcriptional regulation of the salt response of this particular grass, Spartina. And this is actually the data that was actually in 2015. So this has definitely changed, and I should have changed actually. That there are a lot of microRNAs that have been loaded after 2015 until now in the Sanger database. And as I just learned from Nia that the microRNA 820 is only specific to rice, and then might be something in here we don't know. So this is something that we need to look again. But then we do have the microRNA profile of this grass, and then we did a microarray of this microRNA using a commercial facility. And these two microRNA, 397 and 528, actually stood out in their response to salt stress. Okay, and what we did, we actually selected out some of the known microRNAs that are reported in literature to be responsive to salt stress, and these two microRNAs that we identified through our microarray study. And also, so we looked at actually their conservation, among these members, for example, this is in Spartina alterniflora, and then this is in rice, and in corn, and in brassica and Arabidopsis. So we see a good bit of conservation of these small RNA of this grass with rice and other crops. And the ones that we actually saw, we selected out and we looked at their expression. These are the small RNA, their expression in both leaf and root tissues, and also their targets, corresponding targets. Okay, so the corresponding target of 397 here, a corresponding target of the 528. So we looked at the expression of the microRNAs as well as their mRNA targets, so that we can identify a microRNA mRNA module which is more responsive or more operational under salt stress in Spartina alterniflora. And we have overexpressed one of the genes, one of the microRNAs, 397, and we did not see a lot of changes with respect to their tolerance in the very first. But when we actually put this wild type, which was struggling to survive, and then back into normal water, regular water without salt, then the wild type could not make out. It is kind of died, wilted and died. Whereas the salt 397 overexpressors kind of revived and then started to grow. Okay, so this is actually the strategy that we followed with Potatia, because we thought like this is maybe a straightforward approach to identify the genes or to capture the genes that are more direct. So this is actually a strategy, gene hunting. This is not a new technology. It has been used by several groups in several different crops, but not in many plants though. And there have been few reports in plants, but mostly at bacterial level or at yeast level. So we actually adapted a system called COX or FOX, like full length overexpression strategy. So this is actually a functional screening. What you do here is you take the total RNA and then you make the cDNA, double-stranded cDNA, and the full-length cDNA, and then put the cDNA library into actually a vector of your choice, a plant transformation vector, in this case a PMDC. And then you actually make the agrobacterium library with this cDNA library, and the agrobacterium you transform with Arabidopsis or to rice. So this is actually we call gene hunting via functional screening. Okay, so using this strategy, we screened like millions of Arabidopsis seeds. We developed more than 1,000 Arabidopsis transgenics, thousands of Arabidopsis transgenics, and then we actually plated them in salt medium along with the hygromycin selection. So whatever made from here, so these are actually putative salt tolerant plants, and then we actually again in the next generation we picked up these plants, and then in the next generation we again grew them in the plate. Okay, so we grew them in the plate, and we selected out whichever, because some of them were probably escapes, they could not make it, but most of them actually again survived in salt supplemented media. Okay, and then it's whatever survived in the plate, we actually took them into the pot and then again put salt stress. So here you can see that this is your wild type, and then under normal condition they are all growing fine, and when it is salt, then there is obviously growth is very retarded, or growth is affected, but compared to wild type, these plants are looking much better. Okay, and then when we continued that salt stress to the reproductive stage, you can see here that these are some of the lines that actually successfully produced seeds. Okay, so again, like we did a lot of those physiological studies with these plants, looking at their root growth, and then the root biomass, and then the shoot biomass, and the root length, and the relative water content and membrane stability for the the tissue tolerance, at the same time the ion leakage, and also the um, um, the studying the oxygen radicals, or by hydrogen, the hydrogen peroxide, or the pre-oxygen radicals using the DAB or NBTSA. So wherever you see here, wherever you see dark coloration, that means the wild type, for example, here, the dark coloration shows that the wild types are sensitive, and then producing a lot of those pre-oxygen radicals. At the same time, the transgenics are not showing that much of coloration compared to the wild type. So the same strategy we used for rice. Of course, in rice, we did not have the luxury of producing thousands of independent transgenics, but we did have quite sizable number of transgenics. So in rice, we screen them first, and we screen them in float leaf disc assay, and then whatever survived, then we actually also did screen them in the seedling stage. So you can see here, these are the trays that have independent lines of rice, and under hydroponics with 150 millimolar sodium chloride, and then whatever survived, we again went to the next generation to confirm that they are salt tolerant. So you can see here the clear-cut distinction between the wild types here, and then the lines, the rice lines that are overexpressing the Patricia coarctata genes. And again, these are the same physiological and biochemical studies we did to look at the tissue tolerance, and then ion permeability, and also the pre-oxygen radicals and superoxides. So we know that the extremophils not only just tolerate the salt, but they do also have the ability to tolerate the drought, because I think I explained before that even though there is a lot of water, they are growing in a lot of water, but then there is actually a physiological drought because of the salinity in the water. So we also tested these plants for drought stress, and we can see clearly there are few lines that are very, very good in drought tolerance, and then the next generation, when we go, we could see that the wild types hardly could make up after the 2 weeks of drought or no watering, and then some of the lines actually have survived the water stress. Okay, so how am I doing with the time, Shubho? Is it
it's okay, sir. You have a time for.
Okay, all right. So I don't know, maybe around. Okay, so and then once we have these genes, for example, the genes that we have, again, you need to always narrow down. The basic idea is actually to narrow down to focus on only a handful of genes. So we have actually here some genes that we saw for Arabidopsis, for example. The rice one I have not listed here. Again, this is under review, not published. So we actually we took the genes that were in Arabidopsis, in rice, both under salt and drought, and then looked at the interaction among those genes, so it's only those genes interacting among each other. So, and then we saw that these are the genes, some of them I have just dragged them, that these are the genes that have maximum interaction with the other candidate genes. So that actually shows that we probably could target these genes, and ultimately will be perturbing the expression of the other genes that these are interacting with. So that is actually the idea, and we would be pursuing that work. So once we have the genes, we know the genes, for example, in Spartina, so we need to actually functionally validate them, right? So you make a bunch of transformation constructs or overexpressor constructs, and put them into either model plants like Arabidopsis or tobacco or rice. So we choose several genes. I have just given the ones that we have published so far. For example, a case study of vacuolar ATPase. I told you that the plant to be salt tolerant has to have the ability to sequester ion to the vacuole, the sodium ion to the vacuole. And this is actually, the ATPase is actually one of the members of the proton pump, which sequesters the sodium ion into the vacuole at the cost of the proton hydrogen ion, right? And this ultimately also energizes, it creates actually a potential gradient across the plasma membrane or the vacuolar membrane. So it also energizes the other the antiporter genes like sodium hydrogen exchanger, sodium hydrogen antiporter, or the sodium potassium exchanger. So this is one of the key genes, just like the plasma membrane ATPase. We actually saw very clear-cut difference of the plants that overexpressed this specular ATPase from Spartina alterniflora, be it in tobacco or in Arabidopsis, or in rice. So we can see here a clear-cut distinction between the wild types that are actually wilting and drying in 3 days to 1 week after salt stress, and then the overexpressor lines are still maintaining good bit of relative water content to maintain the, to have the good tissue tolerance, and also at this reproductive stage, and these plants, when they were actually moved to the greenhouse, at the reproductive stage, you can see a clear-cut difference between the wild types that do not have that gene and the overexpressing plants over here. Okay, so we have actually a lot of genes. We have overexpressed and then seen some very good response. And this is another gene, ADP ribosylation factor. It's actually a small GTPase family protein, and it's actually more involved in the trafficking. So we saw also that very clear-cut distinction with Arabidopsis and rice. So here you can see also, it's not only at the above ground, but also at the root level. We see very clear-cut distinction between this the wild type and then the transgenic overexpressor lines. And then not only just in salt stress, but also in drought stress. So when we looked at the drought response of these plants, we were amazed to see. And then, well, we knew that the extremophile genes will be also contributing towards drought tolerance, but we did not see that one with the vacuolar ATPase. Maybe it is more specific to just salt stress. But with this ADP ribosylation factor, we found actually very good response both at the above ground and then the below ground root tissues. Another gene, as I told you, that one more ability of the plant has to accumulate a lot of osmolytes, compatible osmolytes. So here is actually one gene, myoinositol phosphate synthase. I know a group of Arun Lahiri Majumdar in Bose Institute. They have done a tremendous amount of work with these inositol pathway genes, and this is one of the examples of that. One of the genes in that pathway, so which leads to the myoinositol biosynthesis. And there also we saw a very good response of the salt stress in Arabidopsis and in vitro and also in the pot level studies. Okay, I would be spending a little bit of time on this particular gene because this is very close to my heart.